The broadcast is now starting. All attendees are in listen-only mode.
Hey guys, all you guys give a big smile to our audience here. Well, good afternoon, everyone. My name is Meng Meng Gu. I'm an associate professor extension specialist in the Department of Horticultural Science. And there were 5 of us got together and we thought that, hey, what would be nice to have a, weekly chat with you all, answering the questions that been sent to us through the previous week and some of the things were going on and stuff like that. So before we go any further, I'm going to ask my panelists to introduce themselves a little bit. Becky, why don't you go first?
I am Dr. Becky Bowling, and my position just changed recently. So this is the first time I get to share my new title. So I am Extension Specialist for Urban Water at the Dallas Center, and I'm excited to be here today.
Kevin?
Howdy, I'm Kevin Ong. I guess a professor and associate department head at the Department of Plant Pathology and Microbiology at Texas A&M, and I direct Texas Plant Disease Diagnostic Lab.
The sandwich eating guy, you. Yankee, why don't you go? Yankee, let Erfan finish. Yankee.
Hey, this is Yankee Joe, and associate professor, extension specialist, a turf pathologist. Happy to join.
You know, I just want to throw in there, before when we had to drive to give different talks, you could at least eat while you're driving. But now that you're doing online talks, I mean, you have like 0 time between things. But, yeah, so my name is Erfan. I'm an extension program specialist in integrated pest management for the Department of Entomology, based out of Overton, Texas, in East Texas, just east of, a couple hours east of Dallas.
Of all the 5 folks here, except Erfan, all the others are doctors, but Erfan is soon to be doctor. Erfan, soon to be doctor. So, yay.
Yeah, we'll see how this whole global pandemic affects my experiments, but I anticipate finishing in about a year.
Okay, all right, sounds good. So basically, soon to be Dr. Erfan Wi-Fi. So we're just gonna talk a little bit about the things that we have seen in the past week. So I'm gonna start this. All right, do you guys see the first my beautiful hand as the background?
Gorgeous hand.
Oh, gorgeous! Thank you, thank you. I was just hoping, just in case this professor thing doesn't work out, I'll be a hand model for hire.
Hand model for plants. Yeah.
Excellent. Yes. Well, for that, I really have to bring up this publication that we just got published. It's this crepe myrtle bark scale, the population dynamics just got published, and with data from Arkansas, Louisiana, and of course Texas. Erfan, Mike Urchin, and myself, along with several other researchers, were on this project. It's 3 years data, multiple locations. So basically, this gonna help us, to kind of predict to any of the degree day models of when we're gonna see a peak of crepe myrtle bark scale crawlers. That again, crepe myrtle bark scale. I want to go back to this picture, and if you are a plant person, you probably realize that, well, this really, just looking at the leaves so fuzzy, it doesn't really look like a crepe myrtle. And if you sense that, you're absolutely correct. What this plant is American beautyberry, Calicarpa americana. It just happened in my backyard. So after I got that news from Erfan, hey, our paper got published, I said, okay, I'm going to go celebrate. So my way of celebration was go back to my backyard, find my American beautyberry, and found a bark scale on that and took a picture. That was my geeky way of celebrating the publication.
So proud of you.
Yeah, thank you. We have been working on crepe myrtle bark scale since 2014. for a long time, really. So we have accumulated a lot of knowledge, and this is one of the paper, and we have accumulated a lot of information on this stopcmbs.com, this website that Erfang is working on. And after my presentation, after this, my slides, I'm gonna, let Erfang be the presenter and, start talking about some of the things that we have available here. Okay, so today I can talk about cream myrtle bark scale for days, but I don't want to bore you too much with that. So today's my topic is going to be a little bit on the landscape CSI. I was walking in the neighborhood, and then I suddenly saw this a flowering pear. It just the leaves looks just weird. The leaves look like this. I mean, they just like clustered and not spreading out. And then if you look, and then I look close to these three sorriest cream myrtle plants. It's like I was just thinking, what's going on with them? Something's wrong. The audience, if you could type in what you think is wrong in this plant, you're gonna get a prize. You're gonna get a surprise prize. So there's a chat box there. There's on the GoToWebinar panel, there's a chat box. So in that chat box, type in what you think that's maybe wrong with this this picture. What's wrong with with the plants on this picture? And you'll get a surprise. You'll get a surprise gift. Okay, so
cream myrtle bark scale on American Beautyberry. Was that the surprise gift?
Of course. Okay, all right. By the way, all the audience has y'all are muted. So if you need a talk at the end, just raise your hand. There's a way to raise your hand, or just type stuff in the question box. So and then we'll notice that. All right. So this is what the Bradford Pear looks like. I mean, all the leaves should be expanded, but they're just not coming out. So you just see these like just the sticks, the sticks with the thread-like leaves. Okay, this is I'm gonna turn to Dr. Yankee Joe. Yankee, you want to talk about spring recovery from the winter turf damage?
So how many minutes I have?
Short.
So I just keep going until the Meng stop me. So I mean, you can notice how turf grass right now growing, because we have plenty rainfall events, and then temperatures going 80, 90, and this is a best time actually the grass start to green up pretty quickly. And many people start to fertilize it, and unfortunately a lot of this is a good time. This is the time that you can see that some of the grass was damaged during the winter, not fully covered, or a kind of slow recovery from the healthy normal grass. So it depends on where you are. I mean, we have kind of weird winter this year, and so we have some several days of cold winter, frost damage on the turf, and so some of the grasses was not fairly uniform, because some of the spot that you can see on the spot, they're still brown compared to the healthy looking grasses. And so I mean, which is common at the frost damage on any grasses, Bermuda grass, St. Augustine grasses, zoysia grass are fairly common to winter damage. And then also during the winter, we got lost by the fungal disease called large patch. Especially we have St. Augustine grasses or zoysia grasses. whether you are recognized is in the fall. Now you can see that the area is kind of damaged. And so the easy differentiations of the winter damage versus fungal damage is you can investigate on the turf surface. So if turf is kind of completely dead throughout the stem, and that's highly possible that fungal disease, instead only like a foliar is kind of melted dry out brown, and still in the stolons are still alive, and so that more contribute by the cold damage. And so at this point, a lot of people are fertilizer to green up fast, and also they consider some fungicide application, but at this point, the fungicide is not very helpful. Instead, you just do some coaeration and put some fertilizer will help to recover those damaged areas. Some of the areas are damaged during the winter will be recovered fairly quickly if the spot is small, and also the stolons or rhizomes still alive, there will be recovered. So it's good you can go investigate it, those patch areas, see if the turf roots or stolons still green and healthy, alive, and there chance that you can recover next couple weeks. And if not, there's no roots and there's bare soils, there's no roots and just completely dead. I mean, again, this depends on the size. If it's small size, it will be like a cover up from the adjacent healthy grass are encroaching and it cover pretty soon. But if it's damaged areas is big, then it's take a while to recover, or you need to have more aggressive approach. Maybe you can implant a new sod in it to make those damage repair pretty quickly. And you can see another interesting pictures on your in the slide on the left-hand side, that you can see the little bit darker green area versus
here. You're talking about here?
Yeah, kind of dark area, which is related to some fairy ring. I mean, there are these areas always have, and usually there it used to have the big tree there, and they cut them down. So the roots is continuously decomposing, and so the other mushroom forming fungi growing start to decomposing the area. So this area is kind of indicate they produce more nutritions by this fairing fungus, and so for now they're green faster, which is good looking, or maybe they reduce the uniformity actually. I mean, is there kind of very, very darker green versus to normal kind of brownish turf, right? So, but there's always chance that when it's the season go, and you can see the mushrooms coming around this dark area, and also susceptible to kind of drought symptoms around it. So that's something that kind of we can see regarding to spring recovery from the winter damage.
Okay, thank you, Yankee. Now, Becky.
Okay, so I wanted to talk a little bit about residual winter weeds today, because this is something that we get a lot of questions about this time of year. All of these weeds that have been growing for several months are starting to flower, go to seed, and they're starting to irritate people. So we get questions from homeowners and professionals alike, what do I spray on this to kill it? Well, The first answer is going to be that this is not the right time of year really to be relying on post-emergence herbicides for controlling these weeds. They've reached maturity at this point, so a lot of those chemical options are not going to be as effective as they would be earlier in the season. If we think about, most of our post-emergence herbicides, they have systemic activity. They move through the plant. As a plant grows larger and it starts to mature, that's a lot more plant for that product to try to move through. And when it reaches this reproductive stage, it's not necessarily translocating things quite as readily as it will at a younger, more vegetative stage. So one of the first things that I would recommend is if you're seeing this in landscapes that you're managing, or if you have homeowners asking you about it, talk to them about the use of pre-emergence herbicide next fall. So we have a lot of people that understand how to utilize these in the spring, and hopefully you've already put one out for this year. If you haven't already, you might be able to still catch some things, but we've already had a lot of things germinate. But putting one out next fall, when our soil temperatures drop to around 70 degrees Fahrenheit for 4 or 5 consecutive days, depending on where you are in the state, this will range anywhere from September all the way well into October. If you manage landscapes in far south Texas, I typically would recommend a sequential application for the fall and winter, because our winters don't get quite as cold down there, so we get a lot of continued germination and emergence of weeds. So you may consider doing your initial application at the timing that we just discussed, and then a follow-up application 8 to 10 weeks later. Make sure that you're paying attention to the product label and you're not exceeding the maximum annual rate in order to achieve this. But for example, a product like Dimension or Dithiopyr that we might put on turfgrass lawns, we can apply that at the high rate at least 3 times within the year without exceeding that rate. We also want to make sure that we're still rotating our modes of action too, even with our pre-emergence. So that's the first thing that we would want to recommend. And, just remember too, that if you're dealing with a lot of broadleaf weeds, like some of these here, we've got burr clover, henbit, chickweed, you may consider adding in Gallery or azoxybin into whatever program you currently have. Those tend to have a little bit more broadleaf weed activity. So if you're dealing with stuff like this in particular, kind of adding that may bolster response. And then just making sure that we get out there and treat those post-emergently a little bit earlier in the season. At this point, I would just recommend that people focus on mechanical control for these mature weeds, and then just kind of being patient. As we continue to get a little bit warmer, we're going to see them start to die back naturally, and then we'll just want to encourage our turf to recover in those areas where those weeds have kind of been sitting during this transition season. So that's the first thing that I wanted to talk about. And then, yeah, the other thing I wanted to talk a little bit about was watering. So I'm already seeing a lot of people out watering quite a bit, and we're still pretty early in the season to really be out watering. I had a homeowner contact me yesterday, and they told me that they've been watering every other day already this season. We don't recommend watering every other day for established turf anytime, typically, because our motto for turf irrigation is going to be deep and infrequent irrigation. We want to water to approximately a 6-inch depth. I know depending on where you are in the state, you might be laughing at that, okay? But we want to the best of our ability, water deeply, and then have enough space between waterings that that upper, few inches of soil begins to dry out, and it kind of motivates our roots to reach for those deeper water sources, and we end up producing deeper, healthier roots in the long run. So typically for established turf, we can meet our watering requirements by watering no more than 1 to 2 times per week, for a total of about a 1/2 inch to 1 inch of water per week. Of course, this should be adjusted if you're getting a lot of rainfall or precipitation, which many of us in the state have been getting plenty of rainfall so far this spring. There really isn't a need for supplemental irrigation right now, unless you're trying to water in products or establish new turf areas. And this is a new guide. I updated it from last year. So it's on the Aggie Turf website, aggieturf.tamu.edu. It's just a simple front and back guide for tips for watering more efficiently, reminding your clients, not to be watering during winter months. We get a lot of people that continue watering all through the winter. This is not necessary on our warm season turf grass, and it's going to promote disease and other issues in the landscape. Really, we shouldn't need to turn on irrigation consistently until usually around May. Then we can keep it on through September, again, adjusting based on precipitation. A lot of other great tips on here, including some discussion around the cycle and soak method to help encourage infiltration, to get that deep watering going, particularly on some of our clay soils. So I just wanted to draw your attention to this and encourage you guys to look at that as well.
I definitely second on the spring rainfall this year. Again, gosh, what is that? I've got so many pictures of daylily rust, and also on the cream myrtle plants, the powdery mildew are spring up. So definitely has a lot to do with the rain. I want to remind everyone that, hey, this is our Facebook webpage, Chat with Green Aggie. But if you search, you have to search Ask Aggie Experts. I think it will pop up. So on that, feel free to leave questions and stuff. Either you're a grower, a landscaper, or anyone associated with the landscape, with the green industry. And this is a group for the green industry. So we discourage any homeowners, master gardeners from joining this group, but this is exclusively for the professionals. Drop your questions here and stuff, and we'll try to answer these, every week. All right, so this is a picture that I took in my neighborhood, and then I got very excited. This thing, so I got really excited. I mean, they're so pretty. They're just, this dark blue, in ornamental horticulture that we're crazy about dark blue stuff if they're flowers. So I sent this to the group, and Dr. Ong, this is what Dr. Ong said. And I actually dropped off a bag of sample for Dr. Ong. He's going to take some SEM pictures. Dr. Ong, take it from here.
All right. So what you're seeing in the photo is slime mold, and slime mold from the group also known as Myxomycetes. There are a bunch of different genera in there. Comes in many shapes and sizes, and what you're seeing is actually part of the what they call a primordial or plasmodial form, where they can move around. More often than not, you'll see them or recognize them looking like dog vomit. So it'll be whites or yellows, and in fact, you want to see a bunch of different colors. Go check out the Plant Clinic Facebook site. That's TX Plant Clinic, and I think 2 Fridays ago I posted a bunch of slime mold with different colors. Now, whites, yellows, reds are pretty common. Blue is pretty rare, but for whatever reason this year, Dr. Gu showed me this picture, and she was either the third or fourth person to show me a blue slime mold this season in a span of about 2 weeks. And so we've gotten reports from this from the Dallas-Fort Worth Metroplex area here, and one other place, I think it was in Harris County, but I cannot recall off the top of my head. But this is really, oh, right. We also got one from the Austin area. So when you talk about, which Myxomycete is this, I don't know for sure, but my guess is this would be known as the Bathamia. Bathamia. That's the genus name. What species? Don't know. I guess you could make a lot of fun and have a lot of fun with that. Now, if you're a landscaper, you probably have customers that say, what do I do with this? Well, you can just use a jet of water and break it up, because what you're actually seeing is essentially the fruiting bodies. Those little things there, they'll dry up, and when you break them open, they'll look dark on the inside, and what those are spores. Spores so that this Myxomycete can continue to propagate and continue on living. Why is it showing up? Well, usually it has to do with the environment. If you got high moisture and then and high humidity type situation at certain periods, you'll see this slime mold showing up in form with this sort of plasmodial structures. They're not gonna stick around for a long time. If you have it real bad, yes, it can smother the grass. So if you're concerned, jet of water, break it apart. If the grass is long enough, get a mower, mow it down short. You know, it's very unlikely that it will kind of return. So if you see it and you want to play along with us on the Plant Clinic Facebook page, go take pictures, post it there. I take pictures of any colors. Um, Now, if you are a nursery producer, uh, hopefully you'll never see this in the greenhouse, but I wouldn't say never. But if you're a nursery producer propagating plants and having young liners, and you might actually have some of this slime mold organisms in some of the mulches that you might be using in your mixes, and if they do come out, there is a chance that it could actually smother that cutting, that young plant. If that's the case, do isolate them, then use the water and blast them off. It's early enough. I think if you do frequent monitoring, that's a simple way to kind of just shoot and wash off those slime mold, and the plants would go on happily. Now, obviously, like with anything, just think about if you put a blanket over a plant, given enough time without the sunlight and the weight of the blanket, you can smother the plant. So slime mold is not a direct pathogen, but the presence of it, it can be a bully to plants. Next, Dr. Gu, what do we have? Oh, roses. Now, did you know, I just got this photos from Dr. Gu this morning. There are two different things potentially going on here, looking at the symptoms. So if you look on the image on your left, which is more of that reddish, oh, and this one too. You see how gnarly those leaves are? We have had a bunch of folks shoot similar types pictures in and freaking out and asking, oh my gosh, is that rose rosette? No, it's not. How can you tell? Well, number one, the leaves look normally sized, and depending on the cultivar of the rose, it may be that reddish color, but the other thing is whenever a plant is stressed, they can have additional production of anthocyanins, which would be manifested as a reddish sort of tinge to the leaves and color. Now, all those twisting is because when the leaves are trying to expand, you got something that is disrupting that cell expansion. And that something in this case is often at this time of the year would be powdery mildew. Early on, you might not see any powdery mildew, but if you go to the next slide, Dr. Gu, on this one, you can actually see some hints of it. Now, it could be a reflection, but where all that twisting is, there's a very good chance when it starts drying out a little bit, flip those leaves over and you will see the fuzzy white stuff on the underside. Now, let's go back to the first slide. Now, that image on the right, we see those this time of the year. And a little bit of those spots, if you notice, it sort of have a tan type a circle. And that's kind of what I call the early incidence of a type of water mold disease. And on that leaf in the middle, you'll probably notice some of the darkest spots that are limited by the veins. So typically when this gets bad, we have what we call Intellinote necrosis. This is suspicious for downy mildew. Roses do get downy mildew when it's somewhat cool and temperature starts to warm up and relative humidity is high. You will see some of this damage occurring. If you start looking on the underside, you're not going to find any mycelia. You're not going to, chances are you're not going to see any of that water mold, of that oomycete. And just to give you the Latin name of that organism is Piranha Spora Sparsa. Why is it called Sparsa? Because it's sparse. So the hard part about this in a nursery type situation, you see the damage there, and you start reacting to it. One of the best ways to basically deal with this is, if you know you have this in your nursery, think about putting in some sort of a oomycete preventative treatment in the future to prevent it when you get conditions that are, I would say, about 60, 65 degrees Fahrenheit, starting to get warm up, and you have high humidity days. The other thing is, if you do get that, then what do you do? If the symptoms are not super bad, perhaps like what you might be seeing in that image, if you keep conditions dry, spread out those spots to allow for better airflow, the problem would sort of dissipate, and while you won't get rid of those damage on the plant, but the new growth would come out just fine. So keep that in mind. If you see this, then consider a preventative program the following year. Once the temperature gets hot, it's very rare that you will actually see this problem pop up. What next? Yay, the other way. I think Dr. Gu is muted. I can see her mouth move, but I don't hear anything.
Can you hear
How
me?
many PhDs does it take to figure out how to work?
Is it better?
Yes.
Yeah, we can hear you.
Okay. All right. The audience said two things. One said it's a virus, and the second answer is a witch broom. And I'm gonna have to say that I will be able to keep the prize to myself. I'll tell you why I don't think it's a virus. Often this virus is plant specific, and in this case, it affects both the flowering pear and the crepe myrtles. So it's probably not virus. Dr. Ong, if I'm off, too much, correct me. But again, I don't think it's a virus. And
Hey, before you reveal anything else, just to let the audience know. So, Dr. Gu mentioned that folks wrote in virus and witch's broom. Just want you guys to understand that the word witch's broom is a descriptor of what you see happening onto a plant.
Right.
The reason for why witch's broom occur, there could be many reasons. It could be a virus, it could be a phytoplasma, it could be chemicals, it could be nutrient deficiency that result in that symptom called witch's broom.
The symptom, yeah.
So but a virus is a pathogen. It's one of several pathogens. So you got viruses, you got phytoplasmas, you got bacteria, you got fungi. So go on, Dr. Gu. Give us the answer.
Well, thank you, Dr. Ong. So for that reason, I don't think this is a virus. And then witch's broom, if you look at this, it's really not. It doesn't really fit the witch's broom, the symptom. In this case, what you want to see is that that these, the twig dying, it spurs, it has a lot of little twigs coming out of these things, that you could see that these, they're like really struggling and stuff. And then if you look at the symptom on these pears, there are some normal leaves, but there are also some of the, the new shoots is kind of like just really struggling. So my best guess, and also if you look at that, all these 4 trees were affected. My guess was some kind of probably it's herbicide, and most likely a Roundup that drift when the homeowner sprayed the herbicide, that it got drift on the branches and the barks and the buds and stuff, and very possibly that and there's certain absorption into the plant body that result in this very severe injury. So if you look at these three, that this one is completely dead, this one is on the way to the deathbed, this one you show some kind of some kind of normalcy on this one. So this is again, I don't think it's virus. Thank you, Brad, but I don't think that was right. And then witch's broom is a symptom discrepancy, and it's not the cause. So Michael, you're not getting the prize either. What do you think?
So, Dr. Gu, is this at your house?
No, this is not my house. My house is much bigger and prettier than this. And how could this be mine?
Oh, I'm sorry.
Yeah. Becky, what do you want to say?
I agree that I think that's a likely culprit. And I think my guess is it could be drift, but it could also be from them trying to clean out these mulch rings. So if you kind of look at the perimeter of the second one from the right, you can see some injury to the turf as well, which I can't see it close enough from here, but it could indicate that if they were trying to use Roundup to clean up within the tree rings themselves, that some of that may have hit the turf, that certainly if that Roundup had come in contact with any part of the tree, that it could be taken up and translocated and create that injury. So that's like a fair guess to me.
Okay. All right. Thank you. So basically you agree with me.
Erfa, do you agree with her?
I mean, do we have any context? Do we know if herbicide was actually sprayed? I mean, I guess that would be one.
That's the first question. Yeah.
That was a great question. Because of social distancing, I wasn't able to knock on the door.
So, let me try and get a sense of this. I got to put it up close to my head to see if the plants tell me anything.
6 feet.
I hear something.
Yeah.
Well, guys, that was kind of a joke. But, one of the things is when you're out there looking and doing part of this, what Dr. Gu called crime scene investigation, I would say, you look for patterns. And you did have some patterns there in terms of the crab myrtle being dead at one end. And then you also have a response on at least one of those trees, the pear trees, showing those type of funky growth on the new shoots. Now, a bunch of things could actually contribute to that. Herbicide is a likely one of the many answers, but also the pattern you have to ask, whether is there a gradient, because those type of symptomology can also be caused by damaged roots. And so with that, is water collecting at the end where that plant is dead, and the only way to find out about that is to ask for progression to the homeowner. Hey, where did you see the problem start first? Now, I'll tell you, many years ago, we saw something similar like that on crab myrtles that died in a row, and it was pretty funny because we finally figured out what killed it, thanks to Atmos coming out to check. Oh, it was close to the station in Dallas. Don't worry, nothing blew up, but it was
Where did Erfan go? Erfan, now you're the presenter. You could go ahead and put on the Erfan, you could put on the website right now.
Yeah, sorry, I had to reboot GoToWebinar for it to allow me to show my screen.
I thought you got scared.
Have permissions and whatnot. No. Do you mind giving me the power? Do I already have the power? Oh, my old me has the power.
Yes, you have the power, Erfan.
Ban the old me and then give the new me the power. Here we go. All right, show my screen. All right. What does it consider my?
Yes.
Okay.
We're looking at your screen.
All right. Excellent. Perfect. So the first thing I want to discuss that I thought was kind of relevant and funny was talking about slime mold beetles. How us entomologists celebrate, in this case, is a couple entomologists from Cornell that discovered some new slime mold beetles. So these are beetles that are associated with slime molds. And the genus is Agathiddium, and they named them Agathiddium bushy, Agathiddium cheney, and Agathiddium musfeldi. It tells you maybe something about their political leanings, named after George Bush, Dick Cheney, and Donald Rumsfeld. So I thought that was kind of funny and relevant there. Something that's been popping up quite a bit is, a lot of people, I think, seeing these. This is pretty typical crane fly season. This is not of particular relevance, say, to the green industry, but you might find this particularly interesting because you may encounter it, or others may encounter it. They're also known as mosquito hawks because they are incredible hunters of mosquitoes by the way they fly. I'm just kidding. That's a misnomer, right? They're sometimes called that. Big mistake. A lot of them actually, as adults, don't even feed. Only live for about 24 hours as an adult, lay eggs, and the larvae are sometimes referred to as leather jackets, and they live under the ground, and they live there for about a whole year, so the whole year cycle until they emerge as adults. So here we are complaining about a shelter in place for a couple of months. These guys have like a full year shelter in place, get to come out one day that year, and then they die. So so far our lives are still looking pretty good compared to that. So I just wanted to cover that there. The Stop CMBS, Dr. Gu had mentioned earlier. There's some excellent resources on there. So if you go to stopcmbs.com, this is specific for crepe myrtle bark scale, where you can come and report any new findings. You can learn about the scale itself and its current distribution. So we did just find, we had a few reportings in Delaware recently, so that's been added to the state list. As well as webinars, and we're starting to get some of the other researchers to start adding some of their data as they go through there. As a grower, I suggest checking out this containerized crepe myrtle bark scale trial because we're setting up a new trial this season, and I've summarized all of my past work, all the insecticides that we've tested, and whether they were great, good, not so good, or inconclusive. So there's some good data there for you to look at there. So that's that. Then I want to discuss, let's see, where did my images go? Boom. All right, so I had a grower that had some, all right, what do y'all think this is? Herbicide damage? Maybe. Witch's broom? Possibly. All right, so it was kind of interesting. So they had several pine trees. It affected only their pine trees, and in two different areas of their nursery that they're getting these pitch tubes, essentially. But if you look at the actual trees, they look totally fine, and have had these pitch tubes for over a year. You look at the rest of the conditions, so they actually had just clear cut a whole lot of pine trees nearby to expand their operations. And so as a result, there's a lot of dead pine trees nearby. So presumably is providing a lot of attractive odors, attractive smells to bring in a lot of beetles to attack those stressed trees. As they're hauling away some of those dead trees, they have a lot of extra beetles looking for a place to lay eggs, get a little bit desperate, and they're going on these pine trees. But since we're not seeing, so I mean, we were looking at a lot of these pitch tubes, and we're actually looking, digging into some of these. So let's see, I got some here. We actually dug in. You can see there's actually no bore or hole. So it's as if the beetle never actually quite made it inside. And these pitch tubes are a defense mechanism of the tree. So usually if you have a very healthy tree, you're not going to have any major problems with these beetles actually causing further harm or actually making it inside, with a few exceptions. We do have historically some records of the southern pine beetle, which, let me see, yeah, here we go. Southern pine beetle, anyone who has heard of the mountain pine beetle, for example, they're in the same genus, Androctonus. It's a devastating beetle to even healthy trees and can really knock them out. So if you're seeing healthy pine trees starting to go south and you're seeing a whole lot of a combination of pitch tubes or little tiny holes of exit holes, it might be another influx of southern pine beetles. There's a lot of monitoring currently going on. The Texas A&M Forestry Service does monitoring for that. And currently they have not found any, I should say in the most recent history, any large populations of them. So they seem to kind of boom and bust. So, they haven't been a major concern recently, but if at your nursery, again, if you start noticing large mortality or lots of borers in your pine trees, please let us know, okay? That'll be a good piece of information for us. So again, pitch tubes, you'll see, and if you dig in, the southern pine beetle kind of has a little bit of an erratic type of gallery. And there's a resource, and this has kind of inspired me while I was looking for a good resource of engraver beetles or just, you know, pine bark beetles in general in Texas. I couldn't find a good resource that kind of had them all and good ways of IDing them. So I'll probably write up a little something with one of our forestry people and post it up on the Six-Legged Aggie website, sixleggedaggie.com. I think a lot of you are either subscribed or part of that Facebook group already. But their galleries are relatively distinct and unique between the different species. So some of the ones that we may encounter here include the engraver beetles. And what's kind of neat about them is the center part right here. Let me zoom in. The center part right here is referred to as a nuptial chamber. So the male goes in there and females are attracted to it. They mate, and each one of these arms is a single female that burrows and lays eggs along the way. And those larvae then create their own little tunnels and galleries. So you can know how many, you know, females are involved in this activity here, and how many you know, groups of offspring basically were produced by looking at the pattern of their engraving. So, you know, by cutting open some of that bark and just looking at some of those patterns, you can get an idea of whether you're dealing with you know, a borer of major concern, or if it's a secondary pest. Maybe your tree is already stressed. If it's already stressed and you're getting borers, you're better off dealing with whatever's causing the stress and making a healthy tree than to just constantly be battling the borers. And that was, I think that was basically, oh, and actually I just want to go back to some of those images. That one grower, there's a good chance that they were dealing with the black turpentine beetle. Again, they will come into areas with high stress trees, or if there are a lot of trees that have just recently been cut down, they will come down, they're attracted in, and sometimes may, as a result, due to that high pressure, go, you know, give a try on some of those healthy trees, but don't always quite make it. So again, here's that tree sap kind of pushing it out. So the tree is good to go. And that's basically it. That's all I got. Do I need to pass this back to you, or can you just take it? Oh, you just took it from me.
Yeah, well, sorry, I just took it from you.
That's all right. I'll try to figure out how to pass it back to you.
I think that's before we you know, ask the audience to, you know, submit any of their questions at this Facebook page or email any of us. I mean, do you all have any other things you want to add? Becky, Yankee, Kevin?
I can add a fun fact about slime mold.
Okay, go ahead.
So this people looks like that similar to the fungi, and so maybe consider some spray fungicide, but they don't like it. But the fact it is, slime mold is not fungi, and it is close to protist, and so it's like algae type. So fungal-like, but it's not actually fungi, and it's a single cell organism, and it's you've probably heard about the amoeba. It's kind of moving around the surface, soils and organic matter. Actually, they're eating the bacteria. They're not eating the plant. They're not eating the dead plant. They're eating the bacteria in the soils. And when they time to, you know, make a spore, then they get gathering in the top of the foliage, which is the turf, the tip of the turf, and they form the spore. So that is not a fungi, and so you cannot get rid of them by the fungicide you have in your stock, and it is the you know, different organism. So
you would say it's a fun guy, but it's not a fungi.
There's a very interesting biology. Actually, they're moving, and those amoeba is invisible, so you can't see it. Only you can see those fruiting structures forming the end of their life cycle.
Well, I
If they're
mean,
amoebas,
obviously,
wouldn't they be more like fungal? Or...
yeah, you guys can see if it's a fun guy, you know, among us all, or a fun person.
Yeah.
Well, hey guys, if you're interested in the Myxomycetes called the slime mold, there's a great book out there by Timber Press.
Okay, all right. Well, thank you all.
Check that out, guys.
It's a nice casual reading.
Yeah. So,
Casual reading. Hey, social
reading.
distancing. got
Nice
time
shelter-in-place reading.
I
Submit
like
your
the pictures in this book.
submit your question at Ask Aggie Experts. Submit it here, and we'll try to find we'll try to get it next time. So again, we'll see you guys Thursday 12:30. And if you have any suggestions, leave it here. Leave it here on the Facebook page, and we appreciate any of your input, suggestions, all the feedbacks. It just it will help us improve this thing. And this is our inaugural chat. This is our inaugural chat, and every attendee will get a certificate of attendance. So thank you all. Thank you all
Check
for
under your
joining us.
seat.
And don't forget, folks, any topics in the industry, whether it's landscape, nursery, or greenhouse, if you have a question, and a burning question to ask about, put it out here. We'll try and answer it. If we don't know, we'll probably point you to a direction to somebody who knows. So thanks for your time, guys.
All right. Thank you. Oh, the Fab
All right.
Five.
Adios.
Thank you, Michael. Hey,
Hey, you gotta put up with her.
I actually like what Erfan did. I think, cause y'all have a whole lot of stuff, and it just, getting stuff ahead of time, to me, it takes certain work. And I think maybe we should just do it as Erfan's. I mean, the transition, as long as we have good internet, the transition and stuff isn't really that bad. So, when you talk from your own computer, you could, just like Airphone, you could talk about so many things, right? What about next time?
Sure, is the PhD student who skewed the curve. Thanks, Airfun.